individual cell in a complex multicellular organism requires a constant delivery of nutrients and a reliable method for waste removal. To solve this logistical challenge, the body uses a specialized connective tissue acting as a centralized transport network. Blood spun in a centrifuge, blood separates into two components. The straw-colored fluid makes up 55%, while the dense red formed elements make up 45%. That top layer is the plasma, 90% water, with a small fraction of suspended proteins. Those proteins perform vital roles. A simple way to remember the three major types is the acronym FGA, for good action. The F stands for fibrinogen, the protein responsible for patching leaks through blood clotting and coagulation. The G is for globulins, which are primarily involved in the body's defense mechanisms, neutralizing foreign threats. And the A stands for albumins. These regulate osmotic pressure, ensuring the right amount of water stays in the bloodstream rather than leaking into surrounding tissues. The plasma acts as a highly calibrated chemical transit highway and the body's first line of biochemical defense. Now, let's look at the bottom 45% of our centrifuge sample, the formed elements. The vast majority of this layer consists of erythrocytes, or red blood cells. A healthy adult has about 5 million per cubic millimeter. They circulate for about 120 days, delivering oxygen before they are broken down and recycled in the spleen. Next are the platelets, or thrombocytes. These aren't full cells, but tiny cell fragments produced by larger specialized cells in the bone marrow called megakaryocytes. This chart shows the leukocytes, the white blood cells. You can remember their relative abundance, from most to least common, with the phrase, "Never let monkeys eat bananas." Neutrophils are the most abundant, actively seeking out and destroying foreign organisms. Lymphocytes drive specific immune responses, while monocytes act alongside neutrophils as aggressive phagocytic cells. Eosinophils exist in much smaller numbers to resist infections and handle allergic reactions. Basophils are the least abundant of all, secreting inflammatory chemicals like histamine. While the plasma manages the chemical environment, these formed elements handle the physical logistics, carrying respiratory gases, patching structural leaks, and actively hunting biological invaders. Blood strictly regulates compatibility, using surface antigens on red blood cells and targeted antibodies circulating in the plasma. This diagram shows the ABO grouping system. Notice the bottom row. Blood group O has 0 surface antigens, meaning it won't trigger an immune attack, making O the universal donor. Group AB has 0 plasma antibodies, making them universal recipients. There is a second major antigen system on these cells, the Rh factor, present in roughly 80% of humans. The Rh designation comes directly from the rhesus monkey, the species where this specific antigen was first observed. If an Rh-negative mother is exposed to Rh-positive blood during her first pregnancy, her body builds specific antibodies against it. In a subsequent pregnancy with an Rh-positive fetus, those maternal antibodies can cross the placenta and destroy the fetal red blood cells, a severe condition known as erythroblastosis fetalis. Beyond identifying foreign cells, blood must also respond to physical breaches in its own vessels. This flowchart details the coagulation cascade. Tissue trauma stimulates the release of thrombokinase. This enzyme converts inactive prothrombin into active thrombin. Thrombin then converts soluble fibrinogen into a rigid network of fibrin threads. Notice how strictly this chain relies on calcium ions to proceed. The circulatory fluid is highly reactive, programmed to defend its borders against foreign cells and instantly form physical barriers to preserve its own volume. Moving from the fluid to the hardware, some invertebrates use an open circulatory system, where blood simply bathes the tissues in open cavities. Vertebrates use a closed network of vessels for precise pressure and flow regulation. The central pump for this closed system evolved over time. Fishes utilize a simple 2-chambered heart. Amphibians upgraded to a 3-chambered heart, with 2 atria and 1 shared ventricle. This comparison illustrates the structural leap to mammals and birds. A 4-chambered heart completely eliminates the mixing of fluids seen in simpler models, maintaining strict separation between oxygen-rich and oxygen-poor blood. The human heart is a mesodermally derived muscle, roughly the size of a clenched fist, protected by a double-walled fluid sac called the pericardium. Internally, it is divided into two small upper receiving chambers, the atria, and two larger, thicker pumping chambers below, the ventricles, separated by thick muscular walls. To ensure blood only moves forward through these chambers, the heart relies on atrioventricular valves. A reliable way to remember their placement from right to left is try before you buy. Deoxygenated blood arriving from the body hits the tricuspid valve on the right side first. Oxygenated blood returning from the lungs hits the bicuspid or mitral valve on the left. This heavily reinforced, valved architecture guarantees a strict one-way flow, maximizing the mechanical efficiency required to sustain a warm-blooded metabolism. Because the left and right sides of the heart are completely sealed off from one another, humans possess what is known as complete double circulation. This schematic highlights the pulmonary circulation. The right ventricle pumps deoxygenated blood through the pulmonary artery to the lungs. It drops off carbon dioxide, picks up oxygen, and returns to the left atrium. From there, the systemic circulation takes over. The left ventricle pumps that oxygen-rich blood out through the aorta, distributing it across an extensive network of arteries and capillaries to every tissue in the body, before it returns deoxygenated via the vena cava. Because the systemic loop pushes blood to the furthest extremities against high resistance, the muscular wall of the left ventricle is significantly thicker than the right. Double circulation operates as two tightly synchronized engines, a low-pressure system to safely oxygenate delicate lung tissue, and a high-pressure system to drive that oxygen to the rest of the body. With the fluid loaded and the pathways established, a single mechanical question remains. What tells this complex hardware exactly when to contract? Unlike skeletal muscles that require a signal from the brain, the human heart is myogenic. It generates its own electrical impulses using specialized nodal tissue embedded directly in the cardiac muscle. Looking closely at the electrical wiring, the process begins here in the upper right atrium at the sinoatrial node. Acting as the system's pacemaker, it autonomously fires an action potential 70 to 75 times every minute. That initial electrical signal sweeps across the atria and relays to the atrioventricular node, positioned near the lower septum. From the AV node, the signal travels through the AV bundle, splits down the interventricular septum, and rapidly spreads through the Purkinje fibers deep into the walls of the ventricles. The heart is equipped with its own internal electrical software, ensuring a continuous, tightly sequenced rhythm independent of conscious thought. At an average baseline rate of 72 beats per minute, the entire sequence of one single heartbeat, known as the cardiac cycle, executes in exactly 0.8 seconds. The cycle begins in joint diastole. All four chambers are relaxed, and blood flows passively from the veins into the atria and down into the ventricles. Next is atrial systole. The sinoatrial node fires, causing both atria to contract simultaneously, forcing a final 30% surge of blood down into the ventricles. Then comes ventricular systole. As the electrical signal hits the Purkinje fibers, the ventricles violently contract. The internal pressure spikes instantly. That sudden spike in pressure attempts to force blood backward into the atria, causing the tricuspid and bicuspid valves to snap shut. This closure produces the first prominent clinical heart sound, the lub. Finally, during ventricular diastole, the ventricles relax. As their pressure drops below the pressure in the arteries, the semilunar valves snap shut to prevent backflow, producing the second heart sound, the dub. The rhythmic thumping heard through a stethoscope is the sound of internal valves slamming shut under fluid pressure. Clinicians monitor this rapid 0.8-second cycle from the outside using an electrocardiograph to obtain a graphical representation of the heart's electrical activity. This chart shows a standard electrocardiogram. The P wave indicates the atria's electrical excitation. The sharp QRS complex maps to ventricular depolarization, initiating systole. The T wave represents the ventricles returning to a normal resting potential. We can also translate this electrical activity into strict volume math. Each time the ventricles contract, they eject about 70 milliliters of blood, a measurement called the stroke volume. If you multiply that 70 milliliter stroke volume by an average heart rate of 72 beats per minute, you get the cardiac output. The heart pumps roughly 5,000 milliliters or 5 liters of blood every single minute. The autonomic nervous system can throttle this rate up or down as physical demands change. But when the parameters of this high-pressure engineering are pushed too far, the system fails. A sustained resting blood pressure over 140/90 results in clinical hypertension, damaging the vessels it relies on. The pathways themselves can degrade. Coronary artery disease, or atherosclerosis, occurs when deposits of calcium, fat, and cholesterol build up inside the vessels, restricting the space for blood to flow. If that restricted flow starves the heart muscle of oxygen, it triggers the acute chest pain known as angina pectoris. If the overall pumping effectiveness drops entirely, it leads to heart failure, often resulting in severe lung congestion. The human circulatory system relies on the precise integration of its fluid medium and the structural integrity of its pathways to sustain life. Maintaining the balance between cardiac output and vascular resistance is essential for the healthy functioning of every tissue in the body.